Cho. Carolyn, can you
Hello?
See if you can share the screen?
How does that look?
Looks good. It's not in full screen yet. All right, so I'm very pleased to have our next speaker again, a very active modelist working in leading industry, Merck, Dr. Carolyn Cho. She is leading the immunology pharmacometrics in the quantitative pharmacology pharmacometrics group at Merck, supporting the drug development from discovery research through post-marketing clinical research. Previously, she had positions as the director of systems biology target group at Pfizer and the global head of computational systems biology at Novartis. Interestingly, she is also a physicist by training from Memorial University Newfoundland, and she did her doctoral research in biological physics at the University of Toronto and the postdoc research in molecular biology at Princeton University. So I want to also mention that Carolyn is extremely active in the scientific community in many positions, leading U.S. federal funding grants and leading working groups of relevant professional societies. And I want to particularly mention that Carolyn Dr. Cho is also a member of the board of trustee of the Mathematical Biosciences Institute. So with that, Dr. Cho.
Thank you very much for the introduction, and thank you to the organizers for inviting me to participate, I think, in this very important discussion. So it's hard to believe after you've seen some of the work that's going on at Pfizer and Sanofi that there's yet another barrier, another big step to actually taking the output of all of this modeling and impacting medicine. So the organizers have asked me, rather than talking about work that we're doing, to give a broader perspective about how that influence has been built historically, not so only from our experience at Merck, but many other companies and participants. I would say I'm not going to talk about COVID. I'm not going to talk about public health. But what I think the organizers wanted us to start thinking about how this work may influence medical practice, and rightly so. The bar to impact how we use, let's say, a new drug agent is very high. And so what do we need to do? How are we doing? How is this modeling actually being used for that purpose? So the first thing I'll say about the application of mathematics in pharmaceuticals, I wanted to point out is that the application of mathematics in pharmaceutical research and development has matured to the point that we have a name. We're called pharmacometrics. We have an academic, if you like, society, the International Society of Pharmacometrics, and we actually, the FDA, the US FDA, has actually a pharmacometrics review division within the Office of Clinical Pharmacology. I'm going to say a lot about the FDA in this discussion. I would like to clarify, as you've heard, that I am Canadian and that there are other regulatory agencies in the world. There's Health Canada, there's the European CHMP, Japanese PMDA, but in fact, it is the US FDA that really is the 500-pound gorilla, not only in terms of staffing and resourcing, but also their legal reach. The FDA, they can withdraw drugs. We can, and it gives us better flexibility in working with them because they'll have a dialogue with us, and They are more willing to make legal commitments with drug developers because they always know that they can come back and withdraw our drugs from markets, et cetera, if we don't follow our side of the agreement. So they have a lot of legal clout. And so a lot of our experience in influencing regulators and medical practice have really come about because of the strength of the FDA. So I'm going to talk about that, but I will also talk about, I hope to remember to talk about the other agencies and what they're doing. So the first thing I wanted to do is to give some perspective about the decisions in drug development from a quantitative perspective. And it's surprising if you're more of a discovery mathematical biologist to hear that the decisions that we're talking about in drug development are highly quantitative. At the end of the day, we want to give advice on a dose of drug, a frequency of dosing, et cetera, and with a lot of additional information that's highly quantitative. And so the mathematician's role here has become somewhat well-defined as we work through this framework of drug development. And these are really kind of categories that I'm going to walk through to try to talk about the influence of mathematics at all of these various levels. The first thing that we're going to talk about is discovery, the right pathway, the right targets. That's really about what is the mechanism of the disease and how do we modulate it. The right molecule, that would be literal drug discovery. The right dose of that drug, as I just mentioned. And finally, what patient population are we actually going to recommend that the drug be used in? And these are all modeling functions. I would also mention, I'm not going to talk about it further, this is not the only application for mathematics in drug development. You just heard about epidemiology, certainly in chemical manufacturing, drug manufacturing, there's a lot of very interesting mathematics, and of course finance, but I won't be talking about that. All right, so I'm going to actually not spend very much time on talking about therapeutics and discovery and mechanisms of action. I think that's been discussed quite a bit here. Most of what you will hear about mathematical biology falls kind of in this category. I would say from a very broad perspective, why we need to do the mathematical modeling and what is impactful for other researchers are two things. The intuition of the empiricist, of the experimentalist, is insufficient to deal with that there's a lot of nonlinear interactions. The mathematical modeling is very impactful when it comes to being able to design experiments to test hypotheses about nonlinear interactions. The second thing is that what we're talking about in drug development is a drug you're going to be taking once a day, 3 times a day. You're going to infuse it for an hour. There's a very dynamic aspect to what we want to be able to predict. Those dynamic perturbations really require modeling. Dynamics is very hard to have an intuition about. So from a pharmaceutical development standpoint, I think that there's this continuing dialogue between experimentalists and modelers. But I would say, you heard in the last 2 talks, there's a lot of data, there's a lot of new experiments coming. People are looking at these illustrative figures here are that we're generating microbiome data, we're generating genetic data, we're generating imaging data. All of those methods, I think, most people accept. We need good modeling to be able to interpret those. Additionally, from a pharmaceutical development standpoint, we're very careful about planning our clinical experiments. We don't want to expose volunteers to a safety risk of an untested drug. And so we're extremely careful in not over-enrolling our clinical trials. So we're always working with insufficient data. I know that kind of goes contrary to what you might have thought, but we're always in a data-poor environment. And increasingly, as we have more data from these other measurements, other experiments, other drugs, we can integrate. But the integration now really does also require a model frame. So this is not so much about influence, but what's coming. I'm not an expert at all about small molecule or antibody development or cell therapies, but the world has changed and it's available now. I mean, it is looking for input from mathematicians. Previously, pharmaceutical companies were chemistry companies. They were valued by how large a literal physical library they had of small molecule structures. So they would have a giant warehouse of 1 million, 10 million chemical compounds, and they would run experiments, high throughput experiments with those compounds. That's really being supplanted now by virtual approaches. So you'll see, for example, previously those chemical libraries really only sampled 10, 20% of the potential chemical space. Now we can evaluate chemicals outside of what we physically have in hand virtually. And then there's been some examples, there's been some really nice work for example, using these generative adversarial networks, for example, to then identify this physical chemical steps of reactions that you would need to make that molecule. So I think here it may be more of a realm of machine learning, and maybe more about physico-chemical modeling, but the structural biologists, the machine learning experts, they are making huge strides here. And there's kind of a revolution in molecular discovery these days because their methodology has been improved by the participation of mathematicians. All right, so now really the meat of what I wanted to talk about is about selecting the right dose. And that's really kind of our role as pharmacometricians. And so taking a step back, how have we arrived at a point where we have actually a fairly large FDA review group that looks at pharmacometrics in submissions for new drugs, new drug approvals? So the first thing is the history of how that influence was developed. There was a group of clinical pharmacologists, probably now 30 years ago, who really themselves learned some modeling approaches and then advocated for those modeling approaches for all the reasons that I've alluded to this point. But what's really influenced our colleagues is that they want to see that what we put out looks the same every time. So it's really boring, maybe, but standardizing how we identify what model we want to build, what criteria we set at the outset to say that that model is good, that the data we're building the model on hasn't been tinkered with, and that we don't make errors in code. We don't make errors in, fixing data sets. We don't do those things because we have set in stone some quality control. So it's very unexciting, but it's really the final step that was needed for pharmacometrics to have those things, so that we are reproducing, our results in a reliable manner. Reliability is extremely important in saying that we understand how our drugs work and how they should be dosed. So 2 case studies, very briefly, that I want to just breeze through. So first of all, I will say that the modeling that we do of clinical data, PD data, sometimes preclinical data, has been used in numerous cases in lieu of clinical trials, particularly in patient populations that we want to treat, but we don't want to put them through clinical trials. The most obvious thing that comes to mind are pediatrics, also for some rare diseases. Also, in cases where it's a little bit more routine, like bioequivalence, if we change a chemical formulation, do we have to run another clinical trial for that? Are there other ways that we can use modeling? I think all of those have been accepted by regulators, not just FDA, but a lot of other countries, Europe. They really encourage that kind of an approach to spare people from having to be volunteering for these studies when we have data that could be understood in different ways with reliable outcomes. So out of all of that has come now the FDA's MIDD pilot program, and I'll say more about that on the next slide. The other example I want to just touch on is that there have been other types of models approved by FDA in lieu of experiments of various sorts. I'm going to talk about this example, but there are other examples, like in cardiovascular safety, for example. More and more there are model-informed safety arguments that have gone in front of regulators. All right, so let me talk about, trying to keep an eye on the clock too, the goals of the FDA's. So the FDA in the U.S. has, and they have an agreement with the drug developers that the drug developers actually pay a fee when they submit their drug application, and the FDA there's a common agreement on how the FDA uses those funds. They've used it to speed up review timelines. It used to be that you'd have to wait 10 years to get your drug reviewed by the FDA. Now it's within, depending on the circumstances, a year to 2 years. So it's been a big change. And it has allowed them now to be able to do things like explore modeling. And can the FDA look at cases that come before them? And I think they're looking to provide a guidance to everybody on what needs to be done in modeling. So this pilot program, as I say, is an opportunity for the drug developers to talk to the FDA about what they're working on, for the FDA to give feedback and to understand what's being done, and to critique it. So right now, the agency prefers to talk to people who are using these models really for very impactful pieces of work. Does the modeling give you an idea about dose that is supported by all of the data? Does the modeling give us a better idea of how to run an efficient clinical trial with a reasonable probability of success? The agency cares that there's a reasonable probability of success. They don't want you to go. They are also protectors of public health. They don't want us to go to a clinical trial that doesn't have a reasonable probability of success themselves. We certainly don't want to do it, and the FDA doesn't want us to do it. So if we can simulate a trial mathematically with pre-agreement about assumptions, that's something that they would like to talk about. And finally, they're also very interested in what we can predict about safety, a much more difficult problem, but they're very interested in that. I see that I'm probably getting to the end of my time, so I'll just mention that the FDA has also accepted. I've just showed some illustrations. It's a model developed at the University of Virginia, and I would refer you to Google some of these folks to take a look at what they've done. They beautiful work, and it's in clinical. It's being used now as part of the artificial pancreas system. Finally, I'll just say very quickly that there is. I want to just touch on this second bullet point about physiologically based PK. This is something that agencies, including Japan, is also very interested. We know a lot about the mechanisms of how small molecule drugs are broken down. There are mechanistic models that have been used in lieu of clinical studies to predict what drug-drug interactions might occur from a particular. So I'm running out of time, so I'll just leave that there. I do want to acknowledge that the sort of machine learning keys, the discovery biology examples that I kind of threw up there, but as illustration, have been the work of many collaborators. And so.
Thank you, Dr. Chow. I just want to give you a hint. Every time when you move your head and to face towards the right angle, actually, the voice is beautiful, is very loud.